Hello, everybody. Thanks for coming to my webinar for the Laboratory Automation and Big Data section of this LabRoots presentation. I'm going to be covering comprehensive genomic association data for genetic variant interpretation, walking through Genomenon's computational approach to facilitate variant interpretation in both the clinic and in pharmaceutical research and discovery. So my name is Mark Kiel. I'm an MD PhD with a background in molecular genetic pathology, and I am the founder and chief science officer of Genomenon, and we provide evidence-based genomic association data to the clinic and to pharma to drive precision medicine. So at a very high level, the subjects that I want to touch on today are the why, how, and what of expediting through big data genetic variant interpretation by beginning with talking about big data applications, specifically in the context of genomics, then discussing how we bring that data to bear to provide value by talking a little bit about Genomenon's flagship product, Mastermind, and how it is driven from an automation and computational perspective by genomic language processing. And then finally, I'll end the presentation by talking through several representative use cases at various levels of genetic variants that are encountered in clinic and in pharmaceutical research. So let me say that Mastermind as a search engine is freely available in its basic edition, and that through this arrangement with LabRoots, you can get access to a trial version of Mastermind Professional, which is used in clinical circumstances and has a number of feature-rich components that help drive the sensitivity and specificity of your evidence search in the clinic. If you just follow that bit.ly link below, mm-labroots 2020, you should be afforded access to the professional trial. But let me begin by talking about big data applications in genomics and set a landscape view by talking to what is needed to fully inform precision medicine studies, particularly in the context of genomics. And the answer writ large is a comprehensive understanding of genomic biology biomarkers and all of the attendant evidence necessary to ensure maximally sensitive and accurate understanding of those biomarkers. And you can break this down into three high-level flavors that are commonly encountered in molecular diagnostics. That is to say, diagnostics themselves, and that is clinical grade, industry standard variant interpretation guidelines to ensure fidelity and accuracy and comprehensiveness of an interpretation, as well as to understand pathogenicity in the form of the empirical studies that suggest what the function of these genetic variants are by individually characterizing each of these variants with the prevailing evidence in the empirical literature. And then finally, the culmination of all of this work is to inform treatment strategies in the clinic or in pharmaceutical research and development by providing all of the documentary evidence necessary to inform some of those early drug development strategies and approaches, as well as toward the end of the drug pipeline, culminating in the design and conduct of clinical trials. So that's what's needed to drive the full culmination of precision medicine studies through genomics. But the challenge is that extracting and organizing and interpreting all of this genetic information requires human intervention to both assemble that data as well as to interpret that data. And that has intrinsic limitations towards scaling genomic medicine. In the clinic, this problem has been referred to as the $1,000,000 interpretation when juxtaposed in the face of precipitously decreasing costs of producing this data in the form of, say, the $1,000 genome, which 5 or 10 years ago was just a futuristic pipe dream. What we're now seeing is that sequencing is commoditized, but the challenges associated with interpreting this data has remained relatively static, absent a solution that we're going to be discussing, like Mastermind, powered by its genomic language processing. Similarly, on the pharma side, there are challenges associated with the human need to organize all of this evidence and preventing efficiency of organization of that data, and also perhaps missing critical components that could unlock future discoveries. And this is exemplified in this quote from a recent review from Nature Genetics that indicates that genetically supported targets double the success rate of drugs that culminate in commercial and clinical success. And so both in the clinic and in pharma, there's a real benefit to having all of this information preordained, pre-organized, pre-annotated, and then allowing the human reviewers of those data to perform the cerebral work of finalizing that interpretation and leaving the muscular work to the machines and the algorithms who are expertly trained in that capability. So some of the specific barriers to scaling both of these different aspects of genomic medicine in the clinic and in pharma are that there's just a lot of information. So there's an information glut, which is a great potential benefit, but absent a streamlined ability to access and organize this information, it becomes a significant challenge. And not only is there too much of this information, but it is largely unstructured, particularly when we're talking about the lion's share of empirical data, which is patterned in the published literature. When we're talking about clinical medicine and the aspects of clinical variant interpretation that are applicable to pharma R&D, there are standard interpretation guidelines to regularize and make more reproducible variant interpretation. But you can imagine how complex and sometimes idiosyncratic those guidelines can be, and how they draw on varied sources of information, which poses its own unique set of challenges. And so the fact that there are guidelines are fantastic, but from a computational perspective, that means that those guidelines need to be adhered to when you're organizing and assembling and annotating this data. And lastly, particularly in clinical medicine, there is the need to have these pipelines that go from raw data or even further to raw patient samples all the way through to this final interpretation. And that requires a synchrony and an integration across multiple different aspects of that pipeline. And that adds to and compounds the challenges that I'm referring to here when we're talking about scaling genomic medicine. So let's talk now about how Genomenon, through Mastermind and through genomic language processing, is surmounting these barriers and rising to this challenge to fully scale and maximize the value of genetic and genomic clinical medicine and research. So this is a new way for us to present the value that we provide to our clinical clients and to our pharma partners. And that is to say, as I mentioned earlier, that we traffic in genetic and genomic evidence, but to put a finer point on it, we surface genomic association data. And those varied associations come in the different varieties that I'm highlighting here in the diagram on the left, where you can see different biological concepts and how they may be interconnected with one another or among themselves. So for instance, a gene and a variant can be certainly tied to a disease, a monogenic or a polygenic disease, whether it be cancer or constitutional, but likewise, it can be associated with a particular therapy or a specific phenotype in the case where a diagnostic disease is not apparent. The phenotypes that are seen in your patient may lead the way toward a specific gene or variant that is the ultimate cause. Mastermind, through its genomic language processing, makes all of those connections and annotates and specifies those connections through categorized keyword annotations of all of those associations as well. And the benefit of having that manifold genomic association network is that the stronger the association, the more likely you are to see multiple of those connections. For instance, there's a strength in finding a therapy that's targeted to a specific disease, but that association becomes even more deeply connected and verifiable when you add a gene or a gene invariant or further when you add additional phenotypes and categorical terms to it. And so those strong associations get even stronger the deeper those biological concepts can connect with one another. And that's the high value genetic insight that Genomenon through Mastermind and its GLP, or genomic language processing, brings into the clinic and into pharma research. So the two ways that I've conceptualized here the insight that we provide being first organizing all of that evidence, again, predicated on the entirety of the medical literature in a way that's imminently searchable and organizable, but then also surfacing otherwise unknown association data. So having the benefit of this comprehensive view and comprehensive indexing of all of that empirical evidence allows us to see things that are otherwise opaque in other platforms or in other users' hands. And so just to be more specific, Mastermind is a genomic search engine powered by genomic evidence that spans the gamut of human disease across all genes and all variants and all variant types across all of genetics, and decorates all of that data with phenotypes, therapies, and any category key terms or custom searches that the user has in their purview. Just to take you behind the scenes a little bit into the software itself, two salient value propositions for Mastermind are its comprehensive reach into the variant landscape of any one of those genes in the human genome, where we can present all of that information in its totality so that it can be searched one by one, or you can look at it as more of a landscape view to see all of that data all at once, but then providing the granular level of information in the form of reference citations at the top right, or their specific sentence context where any of these terms, genes, variants, categorical key terms, phenotypes, etc., are mentioned. You can see how we produce the evidence supporting those automated calls from our GLP data assembly process. A summary of the content in Mastermind is that we include in our offerings for pharma and for the clinical users both population data and in silico models of pathogenicity, as well as the empirical evidence. And all of this data covers gene level, variant level, and disease and drug information as well. And the main takeaway here is that we have the most comprehensive offering of this genetic and genomic information. So part of the intent of this conversation with you is to take you behind the scenes and showcase how we're doing that using the guiding principles of Genomenon's genomic language processing. So bear with me, I have a number of slides that iterate here where I'm going to speak to what that principle is and give you a specific representation of how we employ that principle to develop genomic language processing. So the first here is striking a balance between sensitivity and specificity when you're using computational approaches to data-based automation and data annotation. So sensitivity, I like to say, finding everything you need, and specificity means, and nothing you don't. So being sure that you're not missing anything, but you're also not including too many things that are irrelevant. And the first example that I'm including here is the substrate of your indexing. So what data are you looking through to begin with? And that comes in a couple of flavors here. There's obvious value in titles and abstracts when we're talking about the medical literature specifically. But then particularly for genetics and genomics, there's a rich body of information that's only patterned at the full text level, often in figures and tables, and increasingly with large-scale next-generation sequencing data sets only in the supplemental data. And Mastermind covers all of those different data substrates and does so exhaustively and prioritizes the data according to where and to what extent that information is present in any one of those references. So that's the first measure of the sensitivity in our genomic language processing approach. The next thing is the granules of information in any one of those sources, title, abstract, full text, or supplemental data. And one example here is in our nuanced approach to text conversion. So it's hard to see here, but what I'm highlighting there is a particular variant and the way that it's represented by this individual author, and the challenges that are attendant with reconciling the way that that information is patterned in the actual data substrate itself. So this is a computer's eye view of what that reference was citing, which is to say a specific genetic variant. And there's probably 25 different ways to represent a delta character, which can sometimes be used to describe a specific genetic variant. When this is what a human viewer of that information would see, one aspect of genomic language processing is to put the computer in the place of the human and see the data as it's rendered just as a human would. And so we have developed special techniques that perform character conversion based on optical character recognition by mapping those special characters into a bitmap and then translating that into the most likely match as would be recognized by a human observer. So again, that's talking about sensitivity, making sure that you're not missing those necessary special characters. Another facet of genomic language processing that may be familiar to those of you who know about natural language processing is named entity recognition. Simple examples that I'm highlighting here would be diseases and phenotypes. Some ontologies that capture disease information and phenotype information, ontologies or dictionaries of terms with key value pairs, are freely available to use, including medical subject heading terms for diseases and the human phenotype ontology for phenotypes. And those happen to be widely used across clinical medicine and pharma research. And so when we're talking about the specificity of the search results, we wanted to start where there was the most clinical and research currency. And so we began with those freely available open source ontologies. Similarly, when we're talking about named entity recognition for genes, there is a whole consortia or committee of individuals, geneticists and the like, who are charged with specifically naming these different gene entities with symbols and their representative synonyms. And when we're talking about the entirety of the empirical literature, as we are indexing, we have to take into account previous aliases and nicknames and synonyms. And so we span the gamut in time from retired synonyms as well as current accepted symbols to be sure that we're maximally sensitive. I think the last facet of sensitivity and specificity that I want to speak to is the most complicated, and that is to say how variants can be described. And similar to the HGNC Gene Naming Committee, there is a Human Genome Variation Society which has codified the most appropriate way to accurately name genetic variants. And so that's a fantastic organization who is doing great work. Unfortunately, not every author is faithful to the HGVS system, nor do they always get it right if they try, nor are articles that cite variants from decades ago faithful to those newly codified nomenclature rules. And so the onus is on GLP and Mastermind to be sure that we're not leaving any of those previous or otherwise non-standard nomenclatures unfound. And so to enhance the sensitivity of our search, we paid very particular attention to the myriad different ways that authors can describe variants, whether they're appropriate or less so. A couple of other components here are the difference between pattern recognition and semantic meaning. And our guiding principle here is by starting to just simply identify what's important and leaving the interpretation of what it means to the human reviewers who use our software or otherwise who are internal to Genomenon and develop these very large data sets for our pharma clients. And so we recognized very early that there are a lot of challenges in getting semantic meaning correct, particularly when you're talking about the context of scientific meaning, but that 80% of the value comes from simply accurately recognizing the right entities and then recognizing them in their context. So we've eschewed the need interpret the semantics and the meaning in favor of accurate and comprehensive recognition of the entities and the context in which they appear, and leave the interpretation to the human reviewer. Another facet of GLP is the trade-off between filtration, or throwing away things that you're presuming in your algorithms aren't important, versus prioritization. And so particularly when we're talking about scientific evidence and text and data mining, there's different value that one user might approach the question with than any other given user. And so different filtration strategies may be less appropriate depending on context, whereas a real-time prioritization strategy will allow the user more flexibility in surfacing in real time what aspect of what they're looking for is important to them. And you're less likely to run the risk of throwing something away that's important, which obviously in clinical medicine can be extremely deleterious. Similarly, there's ways to levels at which you can recognize what might be a mechanism to prioritize the data. It may be, say, a specific journal, which has different types of content, a New England Journal of Medicine versus a Journal of Biological Chemistry. Obviously, they have different audiences and different mandates and different areas of focus. And that can be one mechanism by which you prioritize what a user is looking to see. There's also the article level, where you talk about what the content of that article is, which you can infer by the title, by the abstract, and by all of the information content in the full text and supplemental data. And you can shrink that down and make it more granular by talking about the content of a paragraph or even a specific sentence. At the same time, then, that you can prioritize the data based on a gene that you've actually searched for, or related genes in a pathway, or the specific variant that you were looking for, or a widely cited variant that you weren't necessarily looking for in your search, or related variants to a variant that you searched for if, say, there's no information for that search for variant. And so those are some of the different strategies that we invoke in our GLP to prioritize the data that we surface. Additional features that we invoke for prioritization include how many times a term is mentioned, where it's mentioned and next to what additional terms, what context is that citation or that named entity recognized in, what sentence, paragraph, section of a publication or what have you, how well cited is that article, and how recent is that article, or other mechanisms that we invoke to prioritize the data. We always show our work. So there's been a lot of press lately about the opacity of artificial intelligence algorithms and how it's very difficult to penetrate what they mean when you're trying to adjudicate how accurate they are. And our approach has always been, show your work, provide the evidence, particularly in a clinical setting, to justify any scientific assertion that the algorithm is making for the human reviewer to assess with their experience. We also have a healthy balance between the specific bottom-up approach, where we know what we can find very specifically, versus taking a more nebulous general approach, again, using some of those black box AI techniques. We like to focus on things that we know are specific and specifically valuable, the example of which I'm showing there are those named entity variants that we recognize in all of their different nomenclatures. And then again, I want to emphasize here that we have taken the approach of striving to replicate the human curation process, as opposed to taking a black box approach and devising algorithms that don't recapitulate what the human already does, and that therefore would be very hard for a human to interpret the validity of. And so by no means is Genomenon's GLP intended to replace the human process. Rather, it's intended to expedite the human curation process by following their paradigm, whether they're codified in those guidelines that I talked about, or whether they're patterned in the user's own search, but to, as faithfully as we can, recapitulate what a human would do to go through and adjudicate the value of any of that information, but just do so at 100 or 1,000x scale. So the algorithms that we invoke are attempting to be faithful to the human process, just infinitely faster, but in a way that the results can be readily interpreted by the human user. So in the last section of the webinar, I want to go through some representative use cases. The way that I've broken this information out here, these use cases, is by talking about the different types of genetic lesions that our GLP is able to organize and present evidence for, and how that can provide value in clinical and research contexts. So I'll begin by talking about fusion gene data, then I'll talk about another structural variant copy number variants, then I'll talk about smaller variants like indels and single nucleotide changes, and then I'll end by talking about how we can do that database assembly from our GLP and genomic association indexing capability at scale. So let's first talk about the use case of identifying fusion genes. Just as a proof of concept here, we've begun in this approach with the 500 or so genes that comprise the Illumina TrueSite RNA fusion panel. So these are genes that, when involved in a fusion gene event from a large chromosomal disruption, can lead to novel and deleterious biological function that can be treated or can be used for diagnostic purposes. In the case of Illumina, this is for oncology, cancer-related diseases. So for any one of those 500 or so genes, we asked Mastermind, what are the fusion genes that are partnered with each of those driver genes? And we asked how many of those different gene fusion events could we characterize with our GLP, and from how many references did that information come? And you can see the result there on the right, that for any one of those 500 or so input genes, Mastermind, through its GLP, was able to recognize between 1 and 95 unique fusion partners for each of those different genes, which resulted all in, for those 507 genes, in about 2,500 unique gene fusion events. And when I say unique, I mean to say gene A to gene B, where that particular unique fusion event may have any one of a number of different breakpoints, but we counted each of those together as just one fusion event. And all of that information came from the automated indexing of nearly 30,000 articles after sifting through many millions of articles in our database to identify the content that is most likely to be talking about gene fusion data, and that mentions gene A and gene B, no matter what gene A and gene B are, in an appropriate sentence and paragraph and article context that indicates that these are actual fusion genes. And all of this information was automatically assembled using Mastermind's GLP, but then exhaustively manually reviewed to check the evidence, just as I talked about one of the guiding principles favoring the human interpretation over a black box algorithmic assessment. And so this is the takeaway from that work, where I'm comparing in this diagram for each input gene, on the x-axis, the number of unique fusion partners that were identified by Mastermind in blue, or the prevailing manually curated database, Cosmic. And you can see how dramatically Mastermind has outstripped the number of unique fusion partners that were identified using this GLP approach. And the reason that that's beneficial, I'm highlighting here, by showcasing that for this particular data set that we assembled for one input gene, which is NPM1, there was a fusion event that led to a juxtaposition of NPM1 and TIC2 that was not present in Cosmic, but that nevertheless had 9 references that cited its oncologic potential. And indeed, not only was it a driver fusion event, that it drove the large proportion of a particular type of cancer for which there is a targeted therapy. So this isn't just an academic exercise with hypothetical value. There's any one of a number of patients who may have this fusion mutation that, if you didn't have the information for as a clinician, would be lost on you, and that patient would not receive an appropriate treatment. So another type of structural alteration is copy number variance. This posed a unique set of challenges for GLP because copy number variance can be described at multiple levels. Just to orient you what a copy number variant is, it's a sufficiently large section of a chromosome that is either deleted or amplified. And the challenge from a computation perspective is that those nomenclatures that authors can use to describe those copy number variants can come in as many as 5 different flavors without any standardization among them. And so the challenge that we set to ourselves was flexibly and comprehensively recognizing all of the different ways that any one of those copy number variants can be cited, and then automating the assembly of any of that information for any user's input for any type of copy number variant. And what I've done here in this schema for representative copy number variants at the chromosomal cytoband level, as you can see there with 11q23, on a log scale, I'm showcasing how many references citing those particular copy number variants were found by Mastermind as compared to a standard PubMed search, which is the go-to or the previous go-to source for this information. And I'll point out here that the y-axis is on a log scale. So you can see that for each of these, there's at least an order of magnitude more references, and therefore more occasions to find meaningful associations such as therapeutic approaches or functional insight that will allow researchers to divine what's actually going on when one of these cytobands is deleted or amplified in a particular patient. So just like we talked about with fusion, the GLP approach, which is indefatigable and can be tuned for maximal sensitivity and specificity, dramatically improved any manual approach to identifying this evidence. So the next example that I'm going to go through is for single nucleotide or small indel variants. Just to orient you about the meaningfulness of this slide, this is a reflection of the sensitivity of evincing which references are cited from different sources, be they Mastermind or other search resources like Google Scholar, PubMed, or ClinVar. What you're seeing here is for each of those resources, we're sorting by the sensitivity on a per variant basis, where if all of the references that were found in any one of these resources were found by one of those resources, the sensitivity would be 100%. And that's broken down for each individual variant, again, in sorted order for each of those resources. So what you're looking for the quality of the resource is a nice right angle that repletes that rectangle with a maximum area under the curve. And a poor quality resource would be hugging the lower left axis there. And what you can see is that for the vast majority of variants, more than 90% of the variants that were tested in this example of randomly selected clinical variants, Mastermind had 100% of any of the references that were identified by any one of the sources. Whereas when you look at, say, PubMed missed more than 50% of the references in almost all of the variant searches. And Google Scholar and another resource known as ClinVar, which is manually assembled by a user submission, is missing about half of the evidence for any one of those variants that we were discussing earlier, which, as I mentioned before, can mean the difference between a diagnosis and not a diagnosis, or a treatment and a lack of treatment for a particular patient. So the last example that I want to walk through is how we can employ our ability to index and aggregate all of this annotated information at scale to produce manually curated comprehensive genomic landscapes. And this is a diagram that walks through that process. The top part speaks to our computational intelligence and machine learning capability to organize that information, but then it completes by having final expert manual human curation and review. So we have at Genomenon a very unique two-pronged approach where we maximize the potential of computational intelligence, but maximize the benefit of human-level curation, just do so in extremely high throughput. And what one of those genomic landscapes looks like, I'm showcasing here with an example for Rett, where our process was invoked to comprehensively identify and curate the evidence for leading to an interpretation for any one of the variants in Rett as being pathogenic, and even qualifying which of those variants are known to be activating and therefore amenable to treatment. And just like I talked about in our guiding principles, we show all of our work, and every scientific assertion is backed by the source evidence, the source of truth, the reference from which that assertion was made, as you can see here, where that information is all tiered, with the peak being the final call and the base being all of this reference-cited evidence that was manually curated. What that results in, as I'm showcasing here, is a dramatic increase. In this case, it's 3.5-fold, though in many circumstances we go 10 or 20-fold, a broader awareness of disease-causing variants, any one of which could be linked to a patient, any one of which can mean the difference between treatment or absence of a treatment. And we're just exemplifying that in this plot. At the top is the status before, when we were engaged by the pharma client. After many years of manual curation, they were able to identify a couple dozen activating variants. And within a matter of weeks, we were able to increase that by at least 100 variants, which you're seeing there at the bottom diagram in blue. And I like this plot because it showcases the idea that only with a comprehensive view of all of the variants are you able to see salient patterns emerge in the places where these variants affect the protein, and therefore what they may be doing to the protein, and therefore may guide downstream research and development of different drug programs. So this is a summary slide that speaks to some of the genes, just a representative sample of some of the genes that we have curated, and their yield when we produce the comprehensive genomic landscapes compared to the yield that you'd get from a conventionally developed manually curated database, such as ClinVar. So you can see the range there is from a few % more variants that were identified, all the way up to 15-fold more driver mutations with all of the evidence predicate, so that you can appreciate the accuracy of those interpretations. I'll conclude here by emphasizing again that Mastermind, the search engine, is available to you for basic users to guide research for free. You can sign up through that link below, but also through our relationship with LabRoots, you'll be afforded trial access of Mastermind Pro, which is useful in clinical circumstances. And so with that, I'll adjourn, but I'll indicate that you can reach out to us specifically at hello@genomenon.com, or otherwise through the LabRoots event if you've got specific questions about GLP or the content that I've talked about in this presentation. Thank you for your time.